how do the brain cells, the nerve cells, the neurons that connect the motor area of the cerebral cortex down through the brain, down through the brain stem, crossing over to the other side, and going down the spinal cord. Those are called corticospinal motor neurons. My lab is centrally interested in how those corticospinal motor neurons develop. What are the genes that make them develop right? What are the molecules? And the reason we're interested is not just because we want to know how this amazing brain is built, but mostly because we're pretty convinced that we can turn that around to repair those corticospinal neurons. Those corticospinal motor neurons get injured in spinal cord injury, and it's the injury of their axons by crushing, by severing, by pressure that leads to the loss of voluntary motor activity anywhere below the area of that injury. So our hope is that we can learn how to get those guys to be rebuilt, to be regenerated, to be repaired. And I use the word hope, but I don't do a lot of hope. It's actually that we have strong evidence that that's possible. What that means is that in the next 2 or 3 years, we're going to be bringing all these pieces to bear in mice. We already know it can work. How do we get to humans? I think after we have a proof of concept, we're going to need to move to larger animal models. What I would say is it's within rational, reasonable, logical reach. So how is the funding from the Travis Roy Foundation special? Well, it really is, because it enables the highest flying, most venturesome, most ambitious, least conservative parts of our work. What we've done with the Travis Roy Foundation funds, I would categorize in 3 stages, and I can go through those for you now. First, we thought we would approach a question of high clinical relevance in SCI research. If we dream about what we'd like to do, we don't want to make corticospinal motor neurons that all stop in the neck and say, I'm here at the spinal cord, I guess I've found my target. So we undertook a project to identify molecules and genes that make corticospinal motor neurons go long all the way down to the lumbar area that controls the legs, and even further to areas that control bowel and bladder function. And I'm very happy to report that we identified a series of genes and molecules that control and make a difference between corticospinal motor neurons that go to the neck, the mid part of the body, and the lumbar and sacral areas. The second thing we did with the Travis Roy Foundation funding is to say, can we use the genes and the identifiers that the rest of my lab and the rest of the field have identified to find specialized progenitors or specialized stem cells in the adult brain that my lab has already shown several years ago we could turn on and make new corticospinal motor neurons. Using a combination of those genes and molecules, we found a small population of those specialized stem cells right in the adult brain, and now we're doing the experiments to turn them on and try to make new CSMN. So the third thing that we're working on with Travis Roy Foundation funding is we've imported an extremely well accepted and incredibly reproducible model of spinal cord injury in mice. So now we have mice in the lab who have a human equivalent spinal cord injury. Not a knife cut that almost never happens in humans, but rather a compression crush that mimics what happens in people who've had a spinal cord injury. And now we're bringing those 3 things together. the stem cells or progenitors that we think are poised to make new corticospinal motor neurons, the genes that we think can make them new ones and make them go long to the lumbar area, and doing that in mice with spinal cord injury. It's not a matter of hope. It's not a matter of maybe if a miracle happens. It's a matter of doing the work, making it faster, doing it deeper, putting it together collaboratively and inventively to bring the developmental biology to the regeneration of the corticospinal system and regeneration for spinal cord injury. And I think that's going to happen.